So here I sit inside our super creepy prep room to talk to you guys about cellular respiration today. You know, I was just thinking, how does it feel to have my voice piped directly into your ears through your earbuds, like right there in your brain? It's like having Gnant right there in with you. How does that feel? Do you like that? Do you come back? Do you listen to this over and over and over again so you can hear Miss Gnant's voice? I bet you do. That's the nice thing about these flipped videos. Okay, so we're going to talk about cellular respiration today. So cellular respiration, remember, occurs in all cells, not just animal cells. All cells perform cellular respiration. And that's because what those cells need to do is convert that glucose into ATP. Now, it's really easy to fall into the trap of thinking photosynthesis plants, cellular respiration animals. That is not the case. All cells have to perform cellular respiration. Remember, that is one of the characteristics of life, is that they convert energy. So all cells have to perform cellular respiration. All cells need ATP. Most of cellular respiration takes place in the mitochondria, but some of it not so much. So we're going to talk about those differences when we get to them. But you've already learned that sort of connection between cellular respiration and mitochondria, and that for the most part is a good connection to know. So remember, our overall formula for cellular respiration is that we have one glucose molecule, C6H12O6, 6 oxygen molecules, and that's going to give us in the end 6 water molecules, 6 carbon dioxide molecules, and some ATP. How much ATP we get depends on what kind of cellular respiration we perform, because there are a couple different kinds of cellular respiration. Yes, you get to learn about different kinds of cellular respiration. Yay! Okay, so here's the deal. No matter what, there's a step called glycolysis, and glycolysis is always going to come first. Now let's look at that word glycolysis, because you know Gnant loves to break down a word. So glycolysis starts with glyco, and glyco comes from glycose. Glycose is just an old-timey kind of version of glucose. So glycose means sweet or sugar. And then you already know what lysis means. Lysis means breaking things down. So glycolysis literally means breaking down the sugar. So glycolysis is taking glucose and breaking it in half. So glycolysis occurs in the cytoplasm. This doesn't occur in the mitochondria. This is a part that does not occur in the mitochondria. So glycolysis is going to happen up in here in the cytoplasm, okay? Not the mitochondria. So like I said, we're going to break one molecule of glucose in half. Just break it in half. Okay, it's not really that simple, but you can think about it that way. We're going to end up with these 2 molecules called pyruvic acid. 2 pyruvic acid molecules. So one glucose molecule snaps in half, kind of, and you get 2 pyruvic acid molecules. But if you think about what you know happens when something breaks apart, you know that it's going to take some energy. We're not going to have this thing that's just sitting there and it's just going to fall apart. Something's going to have to happen. So 2 ATP have to go in. We need 2 ATP to cause that bond to break. But 4 ATP get created. So 4 ATP get created when that bond finally does break. So it takes 2 to go in, but then 4 come out. Okay, it's like if you say, hey, Gnant, I'll give you 50 cents if you give me $1, and I go, okay, that sounds like a good deal, and I give you $1. You had to give me 50 cents first, but then you got $1 in return. So it's the same kind of deal. Now, I would never make that deal with you. Don't try it, because it's not going to happen. But it's the same kind of thing. So 2 ATP have to go in, but then you get 4 ATP out, which means you have a net gain of 2 ATP. So glycolysis just got us 2 ATP right off the bat. Snap that glucose in half, get our 2 pyruvic acids, we're going to have 2 ATP. The other thing that you know happens is that excited electrons are going to come flying out. And if excited electrons come flying out, somebody's got to grab them. So remember we had in photosynthesis, we had our jar that came along and grabbed those. Well, we have another electron carrier. It's not exactly the same. Remember in photosynthesis it was NADP+. This one is just NAD+. I know it's confusing. I don't name these things. They're slightly different molecules. They're similar, not exactly the same, so they end up with these abbreviations. Just be glad I'm not making you learn the real whole big names of them. Okay, so NAD plus is the jar. It's going to come along and snag those electrons. And then remember, we need to put a lid on the jar. And the lid on the jar is a hydrogen ion. So we're going to get that proton or that hydrogen ion that's going to attach. And we're going to end up with NADH. So we've got the jar with our electrons in it and the lid that goes on top. So NAD plus grabs the electrons, the lid goes on top, and that's NADH. Okay? So when we snap that glucose in half, it takes 2 ATP, but 4 ATP get created in the meantime, and an NADH gets created in the meantime. Okay? Actually, 2 NADH get created in the meantime, sorry. So 2 ATP, 2 NADH get created. Okay? So again, here's glycolysis, here's our glucose. Okay? 2 ATP have to go in, and this guy is going to now break apart into 2 pieces. And then some other conversions are going to happen. And as those conversions happen, we're going to have the NADH and the ATP that are going to get created so that by the time we're finished, we're going to have 2 ATP that we didn't have before. We're going to have our 2 pyruvates, and we're going to have our 2 NADHs. Okay? Don't worry about that other stuff. Okay. So again, one more time. Overall, we've got our glucose, we've got our 2 ATP, and our 2 NAD pluses that are going to go in. And then what we're going to get out is our 2 pyruvates, which are just 3 carbon molecules, 4 ATP, and 2 NADHs. So they're now carrying those electrons. They've got the lids on top. Okay? So that's glycolysis. Glycolysis happens every time, no matter what. Glycolysis is always the start of cellular respiration. Okay? After glycolysis, there's sort of a question that the cell is going to ask itself. Is there oxygen present? We're going to start with, yes, there's oxygen present. So if there's oxygen present, we're going to go on to this thing called the Krebs cycle. And then from the Krebs cycle, we're going to go to the electron transport chain. So we're going to assume that everything is fine and dandy and great, and we've got lots of oxygen present, all kinds of diatomic oxygen just hanging out, ready to go. All right, so here we go into the Krebs cycle. The Krebs cycle is going to happen in the mitochondrion. And the mitochondrion, remember, consists of a double membrane. And the folds of that membrane now are known as cristae. So those zigzaggy folds that are going back and forth are the cristae of the mitochondrion. And then there's this fluidy stuff, kind of like cytosol, that's on the inside of those cristae, and it's called the matrix. So the space between the folds is known as the matrix. If you look really carefully, there are tiny little guys named Neo there. So the matrix of the mitochondria is that space in between the cristae. So the Krebs cycle also is known as the citric acid cycle. And the Krebs cycle or the citric acid cycle is going to happen in that mitochondrial matrix. So in that floaty around fluid inside that space between the cristae, that's where the Krebs cycle is going to take place. So just before the Krebs cycle takes place, what happens is that 3-carbon pyruvic acid is going to undergo a set of reactions, and it's going to become a 2-carbon thing called acetyl coenzyme A, or sometimes we just call it acetyl-CoA. Okay, so our 3-carbon pyruvic acid becomes a 2-carbon acetyl-CoA. Okay, it's just that straightforward. A 3-carbon becomes a 2-carbon. But excited electrons are going to get released. Guess where they go? Yes, you are so smart! NAD plus is going to pick them up, and NADH is going to form. So that proton is going to come on as a lid, and we're going to have NADH. Okay? Also, where do you think that 3rd carbon atom went? Cellular respiration. Carbon atoms, where would they go? Yes, carbon dioxide is going to get formed, and that's going to be your first carbon dioxide byproduct. Okay, so acetyl coenzyme A, remember, is that 2-carbon molecule. That acetyl-CoA is going to go, and it's going to find this 4-carbon molecule that's floating around. You don't need to know what it's called or, anything like that, but it's going to find this 4-carbon molecule that's already floating around. and that acetyl-CoA is going to be all, oh, hey there, how you doing? And the 4-carbon molecule that's there is going to be like, how you doing? And they're going to look at each other and they're going to go, hubba, hubba, and they're going to want to get together. And so they're going to get together, and you're going to get this thing called citrate. Citrate is kind of like citric acid, like this is called the citric acid cycle. So it's kind of like citric acid, but it's not exactly the same as citric acid. So citrate is going to form. Yay! So we had a 4-carbon molecule and a 2-carbon molecule, and they came together to make a 6-carbon molecule. Woohoo! Okay, now at this point, there's going to be a whole mess of reactions that take place, and you don't really care what they are. I mean, you might care, and if you do care, go ahead, find out, research it, watch lots of videos, read your book, learn all kinds of fun stuff, but I don't need you to know all of those very specific reactions. Here's what you need to know about those specific reactions. More excited electrons are going to get passed along to the electron carriers, okay? That means more NAD+, and another new thing called FAD. They're going to come along, more jars are going to come along, they're going to grab more excited electrons, they're going to throw lids on top, and we're going to end up with these electron carriers carrying electrons. So NAD+ is going to grab a hydrogen and electrons and become NADH. FAD is going to grab 2 electrons and become FADH2. You can remember it needs H2 because it's going to grab 2 electrons. It doesn't have a charge of its own. Notice that FAD is just FAD. It doesn't already have a positive charge. So if it's going to grab 2 electrons, it's going to have a -2 charge. If it has a -2 charge, it needs 2 protons to balance out that -2 charge. Okay, that's how you remember that's the one that has the 2 on it. Okay, in that process, 2 carbons are going to get pulled off one at a time. So we've got our 6-carbon molecule. Blink, there goes one. Blink, there goes the other. And they're going to get released as carbon dioxide. Okay, so 2 carbon dioxide are going to get released as a waste product. 1 ATP is going to be produced. And 3 NADH are formed. And 1 FADH2 is formed. Okay, so that 1 pyruvic acid came in, and that 1 pyruvic acid right before the cycle became acetyl coenzyme A, which met up with that 4-carbon molecule. Hey, hubba hubba. And then quickly, bloop, there goes a carbon dioxide. Bloop, there goes a carbon dioxide. And as that's happening, electrons are getting captured and ATP is getting created. Now, if you peel 2 carbons off a 6-carbon molecule, 6, bloop, 5, bloop, 4, look what you've got. You've got that 4-carbon molecule again. But now he's really lonely and has to go back into that cycle looking for another acetyl-CoA to start the cycle all over again. So this molecule is just going to continue doing this where it goes and grabs onto an acetyl-CoA, becomes this 6-carbon molecule, bloop, bloop, and then comes back around and then grabs another one and bloop, bloop, and comes back around, bloop, bloop. And this is all just happening in that mitochondrial matrix. Okay? So as a quick overview again, you have your pyruvic acid that's going to come in, magical things happen, and it becomes acetyl coenzyme A. Acetyl coenzyme A is going to hook up with this 4-carbon molecule, and you're going to get a 6-carbon molecule. That 6-carbon molecule is going to have things happen to it, magical things, where it becomes a 5-carbon molecule, and then a 4-carbon molecule. And in all of these processes, bloop, electrons go off and get grabbed by these various electron carriers. Okay? So overall, we have 2 pyruvic acid, 6 NAD+, and 2 FAD. And we're going to get 6 carbon dioxide, 2 ATP, and 6 NADH, and 2 FADH. Wait a minute, Gnant, wait a minute. You said 3 NADH and 1 FADH2. Where are you getting 6 and 2 from? Aha! Remember that we had 2 pyruvic acids. And so 2 pyruvic acids are going to go through the cycle 2 times. So 1 pyruvic acid goes through 1 time. That's going to give you 3 carbon dioxide, 1 ATP, 3 NADH, and 1 FADH2. And then the other pyruvic acid is going to go through the cycle, and you're going to get all that over again. So for 1 glucose molecule, you're going to go through the cycle 2 times because 1 glucose molecule snapped in half in glycolysis to give you 2 pyruvic acids. And those 2 pyruvic acids then each have to go through the cycle themselves. Clear as mud? Clear as mud. Yay! Are we ready to move on? Okay, so what the heck? We've done all this work and all we've gotten is 4 lousy ATP. We got 2 ATP out of glycolysis and 2 ATP out of the Krebs cycle. 4 ATP, are you kidding me? Plus, duh, where's the oxygen? We talked about oxygen. What happened to the oxygen? Well, hold your horses. Because there's this thing called the electron transport chain. And the electron transport chain is what requires oxygen. Told ya. The electron transport chain happens in the inner mitochondrial membrane. And here's the thing, don't freak out. You already know about the electron transport chain because you know where the electron transport chain already happened was in the light-dependent reactions of photosynthesis. This is going to work almost exactly the same way. Okay, so dude, you already know this. Calm down. Breathe. You've got this. Okay? It's a slow, intensive process. And what happens is it takes the excited electrons from the electron carriers. That's why we needed the Krebs cycle. The Krebs cycle was super important because it excited those electrons. Yay! And got them so excited and got them carried over to the membrane so that this could happen. Those excited electrons are going to bounce down proteins in the membrane. Boing, 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 boing. But remember, as they bounce, boing, 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 they get tired. They're going to lose some energy. And when they lose some energy, they're going to get scooped up by an oxygen and turned into water. So this is where that oxygen comes in, that diatomic oxygen that we needed to breathe in. This is where that comes in, is it's an electron acceptor. It's going to grab those electrons and a couple hydrogens that are around or some protons that are around, and we're going to get water. Okay, so this is where our water byproduct comes from. So the electrons come down, boing, 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 snagged up and make water. Boing, 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 snagged up by oxygen to make water. Okay, but why do we need all this boinging to happen? Do you remember? Okay, so all that boinging is going to happen because it's going to make those protein pumps push protons against the concentration gradient. Okay, and those protein pumps are going to create a greater and greater and greater concentration gradient with those protons. Okay, so now we've got a lot of protons on one side of the gradient, one side of the membrane, and very few on the other side. And when that happens, those protons really, 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 really want to get back across the membrane. And so they're going to look, where can I get across? Where can I get across? Do you remember? Where can they get across? Where can they get across? Not the protein pumps, because the pumps are the ones who are forcing them over to be on that side of the membrane that's so stressful for them. So where are they going to go? They're going to go to the ATP synthase, because ATP synthase is basically like facilitated diffusion for protons. And so ATP synthase is going to allow those protons to go through. But as those protons go through, ATP synthase is going to spin and spin and spin and spin and spin and spin. And what happens when ATP synthase spins? ATP synthase spins and you get ATP, because that's what ATP synthase does. Synth, produce, ACE, enzyme. What does it produce? ATP. ATP synthase. It's the enzyme that produces ATP. Yay! Okay, so we've got this concentration gradient. Those protein pumps are pushing the electrons against the concentration gradient, causing them to build up on one side of the membrane. They want to escape. They're going to escape back through the ATP synthase. That's going to cause the ATP synthase to spin, and that's going to cause ATP to be created. So, again, this looks just like what we looked at with photosynthesis. The electrons are going to come in, boing, 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 and here's ATP synthase spinning because we've got all of these protons over here that want to get through the membrane. And then we've got all of these protons and oxygens that are going to grab these electrons to form water, which is our other waste product. Okay, so the electron transport chain can make up to 34 ATP for one molecule of glucose. 34 ATP for one molecule of glucose just because of boing, 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 spin, boing, 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 spin. Okay, that's going to create 34 ATP. If you combine that with 2 ATP from glycolysis and 2 ATP from the Krebs cycle, then you've got 34 plus 2 more. You've 38 ATP from one single glucose molecule. That's so exciting. So one glucose molecule can give you up to 38 ATP if everything is working appropriately. Okay, so that is awesome sauce. We like that. But that's only if there's oxygen available. What if there isn't any oxygen available? Then what do we do? If there's no oxygen available, remember, glycolysis will still happen first. Glycolysis is always the first step, okay? Then one of two types of anaerobic respiration is going to happen. anaerobic respiration is also called fermentation. Okay, so one of two types of anaerobic respiration or fermentation is going to happen. So fermentation itself produces no ATP. No ATP get produced. So remember, 2 ATP got produced in glycolysis, but fermentation does not produce any ATP at all. Okay, what it does do is free up the NAD pluses so that they can go grab more electrons to keep glycolysis going. So remember, glycolysis happened and the NAD plus came and snagged those electrons. And so what it'll do is take that to fermentation. Fermentation will kind of empty out the jar so the jar can go back and grab electrons from glycolysis again, so that at least glycolysis can keep happening. So if glycolysis is still happening, at least you're getting the 2 ATP. You're also going to get another byproduct though, and there are 2 byproducts you might get from fermentation. One of them is this stuff called lactic acid. And lactic acid is what builds up in your muscles. So if you're running, everything is good, this is great, I feel the wind in my hair and it's fantastic, okay, and you're running, and you've got lots of oxygen getting to all your muscles, and then all of a sudden you push past your limit, and now you don't have oxygen getting to your muscles, and that oxygen's not there. Your cells are going to have to go into anaerobic respiration because they're going to say, you know what, I can't get 38 ATP, but I can get 2. And 2 ATP is better than 0 ATP. So let me do this and let me go to lactic acid fermentation. And so you're going to get your 2 ATP, but you're also going to get lactic acid. And lactic acid is going to build up and cause you to go, oh, I got a cramp in my side. Oh, it hurts. Okay. And so that, if you give it time, your body will clear it away and you'll feel better. Or if you kind of massage it, you can get that lactic acid to disperse and then you'll be better again. Okay. So that's one kind of fermentation. The other kind is alcoholic fermentation. Alcoholic fermentation happens with like yeast. That's how we get wine. That's how we get beer. That's how we get yogurt. That's how we get sauerkraut. So it's just a question of what processes happen inside an individual organism. Obviously, alcohol fermentation does not happen in humans. If it did, people would probably work out a whole lot more often than they actually do. We have lactic acid that says, tells our bodies, hey, stop doing that because we're not getting enough oxygen. Okay? So, running through this whole process again, super quick overview. Aerobic respiration means that oxygen is present. You're going to start with glycolysis, move on to the Krebs cycle, and then the electron transport chain. Glycolysis is going to get you 2 ATP, the Krebs cycle gets you 2 more ATP, and the electron transport chain gets you up to 34 ATP, which means you can get up to 38 ATP from one glucose molecule with aerobic respiration, as long as you have oxygen present. But option 2 is anaerobic respiration. Oxygen is not present. If oxygen is not present, we're going to start with glycolysis again and just head straight into fermentation. We're going to get 2 ATP out of glycolysis and we're going to get no ATP out of fermentation. All right, guess what? That's it. No more cellular respiration. All right, go back, study, learn this stuff. Enjoy.